Okay, good morning. Let's have a class. And as scheduled, we're going to move on to the gram stem today. Okay, so again, we're going to talk about 15 minutes opening remarks, and I'll show you how to do the gram stem, and then you do by yourself. So lab 3, we will talk about the gram stem. So first of all, we talk about the history, and the gram stem is created by Dennis Graham, a Danish scientist in 1886. It is a differentiate stem. And we already talked about in the lecture, based on the color results, we could categorize microorganism, but basically it's bacteria, into two categories, which is gram positive. It will stains purple, or we say it will be blue sometimes. And the gram negative bacteria will stain pink, or we say red. And for fungi, we will say gram staining blue. So we do not really say positive or negative, we just say staining blue. Okay? Now, what are the major steps of the gram stem? And you should be very familiar with that. Is number one, we're going to add crystal violet. And the crystal violet is a basic dye, the function. And you can understanding we are going to put some colors in. Then the second step is gram iodine. And the gram iodine, we call it a mordant, which means we will make the crystal violet stain strong. Then the third step, what we're going to use in is 95% alcohol. And this step, we call it a decoloration. And the decoloration step, it should be short, about like 10 seconds, or these are need 60 seconds. After decoloration, ideally, could be no color. Therefore, we have to add something there. So finally, we add safranin, and the safranin is we call the counterstain. Therefore, based on these four major steps, gram positive, which is purple and the blue, actually is crystal violet color. And the gram negative, we see the pink and the red color, it is safranin color. So this is the major steps. Now we mentioned in the lecture why gram stain positive looks like purple or blue, why gram stain negative is pink or red. We have two theories. Number one is called cell wall theory. The cell wall theory described as gram-positive bacteria has heavy peptidoglycan. So this heavy peptidoglycan will be maintained crystal violet during decoloration process. And in contrast, gram-negative bacteria have a very thin peptidoglycan. Therefore, cannot hold on crystal violet during decoloration, so counterstain showing pink color. The second theory is what we call the lipid theory. The lipid theory, which is described, gram-positive bacteria, the lipids components is low, is about 5%. And the gram-negative bacteria, the lipids component is high, at least 10 to 15% of lipids. Now, part of the reason because of the LPS, lipopolysaccharides. There is a major component is lipids A, therefore their lipids is about 10 to 15%. Because of this higher lipids amount, so we'll say high lipids help decoloration process. So therefore, after decoloration, it's all gone, the color. It's clear, and then you counterstand, showing you pink color. So that's what we talk about is cell wall theory and the lipid theory. Because of the cell wall theory, we need using a very young and we say fresh bacterial culture. Now, how fresh it is, it should be around 24 hours. If it's more than 48 hours to 72 hours old culture, what's going to happen? It will all stain gram negative. And this is, we call it a gram variable stain. The reason is the crystal violet, the major components in the cell wall is already started to cracking down. Okay, so that's the theory behind that. Number 4, it is very important. For the gram stain results, we always know gram positive and gram negative. You will record that, but don't forget, there is 3 major components for the results. So results recording, we should always including 3 major components. Always remember, number 1, gram reaction. Obviously, positive or negative. This is everybody knows. Number 2 is cell morphology. What means cell morphology? The shape of the bacterial cell. It could be cork cell, it could be rods, it could be vibro. Number 3, always remember there's another thing, is arrangement. What means arrangement? How they stack together. It could be single, like E. coli. You already know it's a single cell. It could be 2 of them stacked together. We call it a diprococ cell. It also could be 4 of them stacked together. We call it tetrade. And it also could be 8 of them stacked together. We call it a succinate. Okay, we'll mention about that in the lecture. So this is the 3 important ingredients for the recording is gram reaction, cell morphology, and arrangement. Okay, not only one. So we finished talking about this theory. We're going to tell you how to do it, then I'm going to show you. So what are going to be the steps today? Number 1, you will get a glass slide. Okay, the glass slides, I will do an example with 3 different bacteria, you will do 2. So basically you're going to draw a 2 big circle. The bacteria we're going to using today is E. coli. So write EC on the side. Another one is lactobacillus. So you just write LF. It's a lactobacillus fermentation, so LF. Then don't forget about flip it over. Okay, flip over, and then we will adding a drop of the oil, a drop of the water there. So that's number 3 step is called smear preparation. We add 1 drop of the water, then we add the bacteria, and we're gonna make a big smear. The smear has to be seen. Always remember. Now, later on, we're going to encoding the air dry and the heat fixing. This is the same thing as you simple stem and the air dry 3 to 5 minutes. Same thing. Now, we move on to the major steps of gram stem. And we already talked about that. The crystal, violet, 60 seconds. Of course, you have to rinse. Okay, so we add the rinse, followed by gram iodine, 60 seconds, add rinse. It's a gentle rinse. Then decoloration, 95% alcohol. Remember, only 10 seconds. How you count 10 seconds? You're going to be count 10 Mississippi. Finally, counter stem with saffron. And this is also 60 seconds and rinse. Now, after doing all of these, we let it dry a little bit. And then we're going to using 10x to find sample, and then transfer to 100x with oil immersion. And we want to see 3 important information here is gram reaction, morphology, and rearrangement. Last thing I already mentioned in the lecture, we're going to emphasize here, the procedure you have to follow. The procedure is important. The time is flexible. Which means you may stay in only 45 seconds, that's okay. You stay in 90 seconds, maybe okay, but you cannot mess up with the steps. If you mess up with the steps, the results will be totally different. Okay, so that's a key. So today we will do, they did a good job of E. coli. You see the E. coli, the colony there? And this is a lactofermentant, a very tiny cell there. And this is the first time we do the lactobacillus. It's a fermented bacteria, so we're going to see it. In my lab, I have a Staphylococcus aureus. It looks like it didn't grow very well. We'll talk about that yellow color later on. So I'm going to try this one too. So I will make 3 as an example, and you guys make 2. Okay? So again, we're using one of the steps as an example, then we go back to use observation. Okay? So the same thing, I'm going to have my gloves ready. And then we're using one of the bench as an example, and then we'll do the demonstration to you. So let's, okay, so now everybody, let's go right here. Okay, let's go right here. And we will be moving this here so the other people could see it. Okay, we'll move here. So just borrow your stuff a little bit. These don't move down. And we'll do the rack here. Okay, so the same thing. The first we're gonna check about the water. The water is dripping is good. Okay, I don't know you want to use cold water, hot water. It's cold water. So water dripping is good. Always check. So let it drip a little bit. So we're gonna add it a little bit. And then you have a glass slides. Just take a glass slides. Okay. So glass slides, we're gonna draw 3 circles. I'm gonna draw 3 circles. So it's 1, 2, 3. We'll label E. coli, Staphylococcus aureus, and also the LF. And once you label it, do not forget flip it over, because the marker could be damaged the dye. Now we're going to turn on the fire, the Bunsen burner. It works, and then you're going to have a loop. So make sure those loop is straight. You see, it's broken. So never mind, we'll just trim it out. I'm going to use this one. Be straight. Okay. So we're going to add water there. Before that, you do the sterilization. Make sure it's pink or red. So that means it's burn and kill all the bacteria. Okay, we're going to have 1 drop of the water on each circle. And then we sterilize it again. Okay. So what we're going to do is adding each bacteria. So this first one, we're going to be on the side. We'll pick 2 colonies. What do you call it? We pick some. We are going to pick some big one. So we're going to make an E. coli right here. Make a big smear. The second one, I'm trying to use my own culture to see whether the Staphylococcus aureus is there. Maybe not grow, but we'll see. So I'm going to call down and try to pick some. So whether it's growing. Don't know yet. We'll see. The last one is lactobacillus. This is the first time we do the lactobacillus, so we'll see. We'll call down, and then we're going to pick some colony right here. Hopefully it works. And make a big smear. So what it looks like? It looks like it's all there, so we're going to be doing sterilization of the loop first, and then we do the air dry. Air dry needs to take about like 3 to 5 minutes. After that, we will do a heat fixing. So back and forth a couple of times. I'm just skip that. Okay, directly to do the heat fixing. We'll see what it looks like. We'll directly do the heat fixing. Couple of times. I said it's not cooking. Okay. After that, we're going to turn this off. Then cool down a little bit. Don't touch it on the side right now, because this might be hot. Okay, so we're going to put it right there. Now you have to wear the goggle, because we're going to touch the dye. And the first time you do the gram stem, you may not know the procedure very well, so I will suggest you just arrange it. Crystal violet, number 1. Number 2 is gram iodine. Number 3, decoloration is alcohol. Now, some of you see the acid alcohol on your bench, don't use it, because acid alcohol is for acid faster stain. Counter stain, the last one is saffron. Okay, so we're going to do each of steps. First thing first, crystal violet. So very simple. Add 1 drop, cover the hole. It's not called flooding, okay, just to cover the hole. Crystal violet need to staining 60 seconds. So you can count 60 Mississippi, or you do a timer, that's okay. Then what we do is we're going to rinse. Let's say time is ready, we're going to rinse it. Okay? And then after you rinse, you put a paper towel right here and just dry a little bit. Just dry a little bit, it's fine. Now the second step is add a gram iodine. So we want to make the iodine is strong. So we're going to add a gram iodine. This stays strong. And then let's say the 60 seconds is ready. And what we're going to do is we're going to rinse it. After that, we're going to do the decoloration process. Decoloration, we're going to use alcohol. So that will be 10 seconds. So we're using alcohol. We're going to be doing very quick. We're going to be counting 10 Mississippi, 10 seconds, and the alcohol is ready. The decoloration is gone. So we're going to do the decoloration. And finally, we're going to do the counter stain. Counter stain is saffron. So saffron, we're going to add it right here. And then still 60 seconds. When the 60 seconds is ready, we'll be doing the rings. Okay, after doing the rings, just cover it. Now it's ready. I will tell you, I kind of feeling very obviously, I think that E. coli might not be E. coli because I already see the crystal violet staying strong. So we'll see what it looks like. Okay. Now let's use that microscope. So that microscope may be good to do. So I'll use my microscope to see whether we can find it. Okay, so same thing again. We're gonna practice gram stain using microscope to do the observation, and I need oil right here to do the oil immersion. So let's see what it looks like. So first of all, plug in. Lots of people will forget, and once time goes, if you're in a rush, you may forget about it. So first we plug in, then all the way to the top, and then we're gonna use in 10x. So make sure it's a 10x. And the arrow diagram is at 10. Okay, so we're gonna move to the up to the place you cannot move, and then we do the observation. Okay, I see some of the cluster there. Maybe looks good, so we'll see. Then we're gonna add oils here. At this moment, you could add 3 oil all at the same time. So big drop of the oil. This is not much left, so we'll try to add more oil there. And then we switch to 100x. So we'll see. Okay, very good. I will move this stage to here, and then this arrow diagram to 100, and you guys could come to see what it looks like. Go ahead to see. It is gram-positive, cocktail, and the grape shape. So Staphylococcus aureus certainly works. Come here to see. Can you see it? Very good. Okay, come here to see it. See, I did only about maybe 15 seconds. The time, it doesn't matter. Come here to see it. I think it's a very good picture. It's better than lots of the textbook when they put there. The cocktail, it's seen, and you see the grape shape. Okay? So that's the Staphylococcus aureus, and I need to tell my students this one we may need to be restrict. Ideally, it's good. I'm going to move to E. coli to see what it looks like. Okay, maybe works, maybe doesn't work. So what you see here, surprisingly, this E. coli seems it's gram-positive.
Oh wow!
But it looks good. It's large and short. So come here to see. Then maybe it's not E. coli. Is that right? Because E. coli is supposed to be gram-negative. We'll see. Come here to see it. Looks good? Yeah? Okay, now let's see lactobacillus. That's the only question. Usually you go the other way. Is gram-positive bacteria become gram-negative? That could be because gram variable still. We'll see the last one. Okay. The last one, what it looks like. Surprisingly, The lactobacillus is also looks like a Staphylococcus aureus. So let's see. Let's come here to see it. And that's fine. We'll see what it looks like. Okay, I did it. It looks like Staphylococcus aureus, but I have a 3 pathogen, so maybe I'm got a cross contamination. So you're gonna have to verify it. Ideally, E. coli and the lactobacillus looks like the same large. E. coli is short, lactobacillus a little bit large, but one of them gram positive, another one is gram negative. So you decided. Oh, is that okay? I'm already showing you. So come here to get your plates. One lactobacillus, another one is E. coli, then do the gram stain. We'll see what it looks like. Very interesting, the results.